This technique can help answer key questions in the cellular infection and microbiology field, such as how hemocytes contribute to the overall immune response in fruit flies.
The main advantage of this technique is that it can be performed laboratory using previously infected larvae. Alternatively, hemocytes from uninfected larvae can be used for ex vivo infection.
Visual demonstration of this method is critical because the imaging steps are difficult to learn because of the complexities of the confocal microscope and the options for 3D model reconstruction.
To begin the protocol, layer 2 to 3 pieces of 10 cm by 10 cm paraffin film under a stereo microscope. Next, prepare the glass capillary by setting the capillary puller heater to 55% of maximum. Pull the capillary tube to a sharp point of approximately 10 micrometers. Backfill the capillary with mineral oil. Assemble the filled capillary tube onto the nanoinjector and open the fused capillary tube tip by breaking off the tip with forceps. Eject as much oil as possible from the capillary tube tip, then fill the tube with Drosophila hemocyte isolating medium, or DHIM. For easy visualization of the border of hemolymphin oil, include an air bubble between the oil and DHIM. Using forceps, gently pick 10 third instar Drosophila larvae from the inside wall of food vials